Hi everyone, this is Dr. Santosh. Today I'll be talking about factors that are involved in heterochromatin formation. So you can see here, these are the factors that are involved in heterochromatin formation. The first factor is DNA. Okay. Second factor is related to histone proteins and HP1 protein, heterochromatin protein. P1 means heterochromatin protein. Now, and fourth factor is nuclear RNAs. So these are the four factors that are involved in heterochromatin formation. So first factor, when in DNA there is a large array of tandem repeats, the same repeat when they are repeated many times. Okay, so such DNA sequence favor heterochromatin formation. For example, this type of repeat you will see in centromere and also in telomere. So this type of repeat, which is in tandem repeats, the same sequence repeated many times in tandem, they are called, this type of repeats at centromere and telomere, they are called satellite sequence. So these sequences favor heterochromatin formation. Other type of satellite sequence is mini satellite. We also call this mini satellite as VNTR. What does that VNTR means? Variable number tandem repeats. This variable number tandem repeat sequence also favors heterochromatin formation, and other very highly repetitive sequence can also influence heterochromatin formation. Now, second factor in DNA related factor is methylation. So when the DNA, especially methylation in DNA, happens at only one place, when there is cytosine and guanine. This is P4 phosphodiesterone. When you find this type of sequence, cytosine guanine, so methylation in DNA will always happen on the cytosine. So cytosine methylation, there will always be cytosine methylation. In such sequences, you will find methylation. And when some part of DNA which is methylated, that part of DNA is liable for heterochromatization or heterochromatin formation. The best example is imprinting. So in imprinting, you will find that methylation of DNA, and that DNA will be heterochromatized. Now, second factor is histone. This is a very famous, very important factor. You will see histone modification, and especially histone modification, methylation of histone will cause heterochromatin formation, and hypoacetylation, deacetylation of histone will cause heterochromatin formation. And there are some alternative forms of histone. You can see here the CNPA. This is centromeric histone. This is variant of S3 histone. So this S3 histone variant and this H2 variant, that's macro H2A. This is generally present in bar body, and this is in centromere. So this variant of histone, they favor heterochromatin formation. We have seen that this type of histone we have found in heterochromatin region. So there are alternative forms of histones. These are alternative forms of histone that actually stimulate heterochromatin formation. So histone methylation, it's very famous. Histone methylation, especially histone 3 and lysine, 9th lysine in histone 3. When it is methylated, it will facilitate heterochromatin formation. And for bar body formation, S3 lysine 27 trimethylation will promote heterochromatin formation. Now, histone deacetylation is very famous. When the histone is deacetylated, it will lead to heterochromatin formation. And then after this histone has been methylated, especially, now the protein that will help this histone methylation is HP protein, heterochromatin protein, and that protein will associate with this methylated histone and will cause heterochromatin protein. So the HP1 is a very important protein that will cause heterochromatin formation. Now nuclear RNAs, especially when you really want to know about nuclear RNA, you need to know the nuclear RNA related to bar body formation. There are two nuclear RNAs. One nuclear RNA is called GIST. What does that mean? X inactivation specific transcript. X inactivation specific transcript. This GIST is nothing but long non-coding RNA. It is a long RNA which is non-coding, does not code for protein. And this long non-coding RNA, this RNA is responsible for formation of bar body. This bar body is a heterochromatin X chromosome. Now, other long non-coding RNA which is called psi X. This psi X opposes this. Psi X opposes this zest. So this is also long non-coding RNA. So this long non-coding RNA is very important. This nuclear RNA is very important. It plays very important role in heterochromatin formation. So thank you for this class. I hope I made things clear. See you in next week.